Okay, this lecture will focus on continuous manufacturing, and at the later part of the lecture, I will also touch on some of the modeling aspects related to pharmaceutical systems. But let's start with the continuous manufacturing. And the idea for this first part of the lecture is that we compare batch processing with the continuous manufacturing concepts. And here in this lecture, I will highlight the importance of process analytics and process control in the context of a continuous manufacturing setting. And we will also discuss some of the innovative manufacturing solutions of the future products. They can be based on additive manufacturing or 3D printing, if you will, and I will show you a few examples of those. And then I will finish the lecture by discussing some of the aspects related to modeling of pharmaceutical systems. So it's important to increasingly use computer predictions for understanding how systems behave. And in the context of manufacturing, this is important both at the bulk level and at the molecular level. But let's start by thinking about what is continuous manufacturing. So there's one important concept in the pharmaceutical world that is often affecting our thinking. It's the concept of batch. So batch processing and definition of batch is important for pharmaceutical products. And it's defining the related to quality systems. And it's related to many aspects. So our starting materials, they have a certain batch number. And then when we are producing the product, then we are doing batch documentation for a given batch. And then we package, put them in a packet and have the labeling so that we always refer to certain batch number where all this documentation is related to. And one day, if we need to do a batch release, or we are seeing an auto specification or even product recall situation, then we are always referring to a batch number. And this is very problematic if you think about continuous processing. So continuous manufacturing is referring to a scenario where we are producing constantly, all the time, the same materials. So then this definition of batches is a bit difficult. But I will come back to it, how we can also survive that situation. So then important is basically just follow the time, and the batch release is becoming a more like moving item. So the time point of production is the identifier for certain part of production. Just a flashback reminder. So this is the legal framework for doing GMP. So many of these GMP chapters, they are describing different parts of pharmaceutical quality system, and the concept of batches is quite deep in here. So many of these parts are referring to the batch concept. So when we move to continuous, then we have to be a bit creative in there on thinking. And let's see what this really means. So here is an example and a contrast with batch production with continuous manufacturing. So if we here think about typical batch processing, you start with your raw material, then you keep on processing until certain time point, 4 hours here in this case, and then you isolate the product, and then the batch is finished. So this, whatever 4 grams or 400 kilograms, so this is one batch of material. And then this is the cycle how you do it. But in the case of continuous processing, the idea is that you have a processing environment where you feed in the raw material all the time, and then you at the same time you remove the material out. So you reach an equilibrium here. So then your raw material feeding rate is matching with the product removal rate. So then when you reach that equilibrium, then you can feed in material and take then material out in the same rate, and then you can keep on processing 24/7. So then you basically produce all the time, and that gives a certain benefit for the whatever productive industry. So you can make more efficient production cycles and smaller production geometries. So there are many benefits from this approach. And if we think about how it's done in the pharmaceutical field, so this is an example of a batch manufacturing. So there are different unit operations. You have a milling unit here, and then maybe some granulation tools, then tableting and coating units. And when you are making your one product, then you do it with this type of steps here with the different batch processes. Or if you have another product, then you produce it with a different setting. But the idea with continuous processing is that we basically remove all this and get them connected so that material can be handled as a continuous flow of material. So it starts with a continuous mixing of powder, continuous granulation, tabletting can be continuous, and coating process can be continuous. So in this case, then we are connecting all these processes, and then we have basically this tube, this imaginary tube that is used for processing material. And this is an example of again, this batch processing. So typically it involves testing and storage after each step. And then this really reaching the final coated tablet, it can take several months. So if you, for example, produce your API in some other part of the world, and then you need to ship it to the unit where you are doing the tablets. So then all of a sudden we are talking about months here. So indeed it would be more efficient that everything is happening in the smaller geometry, and then you do the whole flow continuously. And this is still a conceptual idea of doing this integrated continuous manufacturing, end-to-end manufacturing of tablets. And there are a few examples out there. They are leading academic units and leading academic players working on it, but it's not very magical. So same solutions have been implemented to other industries. So if you think about how pulp and paper industry is operating, so we are still making some paper and every now and then these machines, they are naturally continuous processes. And if you think about the paper machine, so this is something where you're feeding in the trees from the other end, and then processing very fast, even several kilometers a minute can be handled in these paper machines. And then that is very large units, and something that contains water to high content and then also low content. And then this is to some level very similar to tablet production. So we have cellulose in the tablets, and then we have wet granulation, and then we want to dry it. So some principles can be taken from other industries. And this is indeed how the modern tablet or this pulp and paper line is working. So you have lots of measurements. So these process measurements are bringing the possibility for controlling this in this speed, and then having always a desired quality. So the future pharma production is moving to continuous processes. So we have this basically imaginary production line that is then feeding in chemistry from other end, and then getting the tablet out from the other end. And some of the parts of this are already continuous. So if you think about, for example, processes like dry granulation, so we are feeding in here the dry powder and then compacting it here between these rolls and then grinding it here. So it looks very much like a continuous process. So this is one example of continuous processes. Extrusion spheronization is another process that is by nature, it is continuous. So these extrusion processes, they can be handled continuously. And then this is an example of a commercial continuous production line that also contains tablet machine. So it's a commercial instrument line called Consigma from GEA. And then here we have a granulation tools and a drying unit, and then feeding to tablet machine in the same production line. And this is all working continuously. There's also an interest to move towards getting all these aspects into continuous processing, and then also having the continuous synthetic reactions. But this still requires some engineering and indeed some modeling simulation, and in order to make it happen. This is one example of those activities. So this is a team from MIT, and then together with Novartis, they have been working to end-to-end continuous manufacturing line that is integrating synthesis, purification processes, and final dosage formation. And in their solution, this is just a technical drawing of it, so we are not going into details of all these processing steps. But what happens here is a different type of chemistry is being handled in different steps. So we have a part of reaction happening here, then maybe some separation process here, another synthetic reaction here happening stepwise, and then finally we reach some sort of roller compaction here that is then extruded and then maybe melt extruded here with the PEG, and then out comes something that is an injection molded tablet. So the idea here in their work is that they are having the integrated end-to-end continuous production line. And one important concept in such system is then the time aspect, as I mentioned in the beginning. And a very important parameter here is a residence time distribution. So then you need to start thinking about how much time your material has been spending in this process. And one theoretical consideration here is an imaginary scenario where we have a plug flow. So the material is really moving as a plug and then not being distributed in the process. But in real life, there will be distribution of material in there, and that can be theoretically handled by using continuously stirred tank reactor thinking. So we basically imagine that the moving of material is like smaller reactors. These are imaginary reactors that are connected to each other, and then the material is moving here. And then depending on number of these reactors, then that describes how much the material is spreading in this production geometry. And this maybe becomes more visual with few examples. So we can imagine here a reactor. So that is a continuous flow reactor. And then we inject some material here, and then we detect it at the end of the reactor. So you can see that we can do either pulse injection, so pulse like this, so concentration increase very highly here, and then it disappears, and then you look how this concentration is then distributed over time, and this would be then the residence time distribution of your material here in this production geometry. You can also do something like step injection, so you have a step increase in the concentration of an analyte, and then you see how fast that step is going through the process. And then you can mathematically calculate the residence time distribution from this signal. And this can be visualized in many different ways. So this is one way of presenting this pulse or step change or variable step change. So then you can see that in the time point 0, then you have either this pulse going in or step change going in, and then you see how it's being distributed in the production geometry. So with liquids, these changes are happening faster, but with powders, this diffusion is slower. So then it's also mainly mechanical dispersion of powder, and that's then something that you need to experimentally investigate. And this is something that can be analyzed, for example, in the twin screw powder feeder geometry or hot melt extruder. So they are the same principle. And then what comes out here is a similar type of signals. So we can look into the step change here and then the output of that. And then we have an estimation of how things are happening in this given production geometry. So how fast does this change come through the process? You can also do similar type of step change in the process. And in order to measure this, in order to understand it, you need to have, again, these PAT tools. And spectroscopy is always a lovely solution for looking into concentration of analyte in the moving material. And here is a visual example of a bit longer production line. So what we are having here is a powder feeding with a loss in weight feeders. So we have loss in weight of excipient, loss in weight of API, and they are being fed into a continuous blender. which is then blending, and then we have NIR measurement of that blend, which is then being fed to a roller compactor. So we are doing this dry granulation, and then we measure again the properties of that, and then finally we make a tablet, and then collect also the tablet concentration. So you can imagine that the step change here, it involves many different production phases. So then it will be start spreading broader and broader if you are doing a change here. So if you start something that is very sharp change in the feeding system, that will start diluting, so to say, in the process. And then you can, with these real life examples, you can see that all of a sudden quite dramatic changes can also dilute into when you look into the final tablet. So then you can see that the big change here in the beginning is then a bit smaller change here for the tablet concentration. But then if you start seeing these changes, so then you need to specify and make a specification for your product, and then how much is acceptable. And then if you start seeing the sliding towards strong quality, so then you know how much it has been in the process. So then you can start estimating which part of the continuously manufactured product needs to be discarded. So if you know the residence time distribution, so then you know that approximately 40 seconds of production needs to be discarded because of this problem. So that's why it is important to know the residence time distribution. And again, these installations you can do differently. So most likely this inline installation would be very useful for really measuring the properties of material while it's moving in the production stream. And we can look on an example a bit more detail with the roller compaction. And so roller compaction is a process where we are feeding the powder here and compacting it with these counter-rotating rolls here, and then we are making something called ribbon, and then that looks like a piece of paper made out of excipients and API, which you then push through this sieve, and then you can get the granules, which you then compact. So this is by nature a continuous process, and then you can imagine that it would be useful to measure powder properties from this ribbon and from these granules and finally from tablets, so that you can get an idea of the quality of these tablets. So here we have a visual illustration of a cycle of this type of work. And this is an activity that we have been doing here with one of our students. So we made a simple roller compacted formulation consisting of cellulose and acetylic salicylic acid. And with this setup, we investigated the effect of roll pressure and roll speed on the properties of ribbon, granules, and tablets. So we can do with near infrared, we can measure all these products and then see some characteristic features. And since we are now doing design of experiments, we are changing systematically these process parameters. We can see how our design space is being affected by these changes. And in this specific case, we had also interest on the ribbon porosity, amount of fines, and granule particle size. And then we can start seeing that how these parameters that we were changing, so roll speed and roll pressure, how they are affecting porosity and fines and size of granules. So we can see that roll pressure is decreasing porosity and then decreasing the number of fines. And then in the same way, it's also increasing the particle size. So this DOE results can help you in understanding how changing process parameters will affect the properties of the granules. And this knowledge is then something that we should translate into the use of PAT instrument. So we would like to measure these parameters, and then this is a summary of these findings. So roll pressure and roll speed have a significant influence on the responses, on these responses. And then these we can then control by measuring with NIR these properties. And for that purpose, we are doing something called chemical imaging. So instead of measuring only one spectrum, we are actually doing a mapping of our samples. So we are taking a chemical photo of our sample, and with a certain resolution we can measure many different spectra. So we can, in this case, measure 6 times 636 spectra from this area, and all of them can have a different composition. So then we can start looking into how homogeneous our sample is, and then also the analytical signal from each. So this is a bit extreme example that is very segregated samples. So the component 1 is here in this corner, and component 3 is in this corner, and component 2 is in the middle of our sampling area. So that is indeed quite extreme. But then you can imagine that depending on a sample, then you would see potential in homogeneous areas or some other factors that are being affecting the NIR signal. There are many physical factors that affect these spectroscopic measurements. So we are doing non-contact measurements. So we are not only seeing absorption of light, but we are also seeing some other phenomena like reflection. We might see transmission of light, and then different scattering and reflection type of information, and even fluorescence. So this means visually something that your light is coming to the sample, and then it can directly reflect. So this has no chemical information. So what we would have interest is maybe a transmitted part of the light. So if you can measure, for example, in case of tablet, if you can measure transmission measurement, that would be useful for telling you what is inside the tablet. So many of these measurements, they are happening in the scattering mode. So then you have a diffuse scattering, and then the signal that we collect with NIR is sum of all these components. So the signal that you detect contains both chemical and physical information. And this can be measured with this different type of NIR imaging setup. So this is an example where we have a conveyor belt here where the sample is moving here in this direction, and a line camera here that is presented here. So we measure one line here and the sample is moving, then we measure second line, and all of a sudden we can make pictures of objects. So this is fascinating because you can do it quite fast. So we can measure several centimeters a second, and then if you measure 10 centimeters a second of this quite broad conveyor belt, so you can see that you can take a chemical map of hundreds of tablets in a minute. So that would be quite useful information for continuous production line. And in this specific case, we were working with cellulose and acetylic salicylic acid that have a quite nice spectral signals. So excipient is very different from the API. So our model API is having a unique signal here. So what we could see is that depending on compaction pressure, so the mixture of these two components was also changing. So we can indirectly measure the porosity of the samples from different compaction pressures. And then that is very useful for building up a full picture of the material. So when we increase the compaction pressure, then we have a less porous ribbon, and that would affect the spectral baseline. And in this case, it was very useful information for measuring the porosity of these samples and building up a control loop for that. And this is a well-known phenomena. So here's an older paper showing the example of increasing tablet compaction pressure on the spectral baseline of NIR spectrum. And this is something that you can also consider for controlling a tablet machine. So in our specific case, what we could do is then to do this chemical mapping. So we can look the porosity of ribbons, and then also make a chemical map at the same time. So what we can see here that in the different roll speeds, all of a sudden we don't have a uniform porosity of material. But at the same time, we could see that we had quite uniform chemical composition, but then the physical composition was changing. And that can be then problematic if you link it to the behavior later on. And in the same way, we can take a chemical map of tablets after compaction and then make sure that we have the right amount of API in them. Yes, so I think this might be a point where we need a short break. So let's take again a 5-minute break here.